Hello and welcome friends. As you all know that in this session of ours we basically cater to the science subjects and today also we are going to discuss on a very interesting topic. You might have heard about antibodies. Today we are going to discuss on antibodies. We are going to discuss basically the structure as well as function of antibodies. What are the various types of antibodies, what are its major functions and how they protect us from various kinds of diseases. We are going to discuss all this in this session of ours, and for this we have again with us in our studios Dr. Amit Bhattacharya. He is Assistant Professor in Ramjas College, University of Delhi. For him I would like to say that he has immense knowledge as well as immense experience. As far as his experience is concerned, he has contributed almost 6 years in the field of teaching as well as he has a vast research experience of almost 8 years. So first of all I would like to welcome our guest Dr. Amit Bhattacharya. And sir, as through previous sessions also, through you we try to discover and learn lots of new things. Today also I hope that we are going to get a deep insight into this topic.
Yeah, thank you very much, ma'am. So today I will take up the topic of antibodies, and I have segregated the topics into various segments. The first segment I will focus upon what is an immune system and how does this antibodies contribute to this immune system. Second, I will go into the different discoveries which were made in this antibodies field, how these antibodies were discovered by the various scientists, and subsequently I will go into the various types of antibodies and what exactly are the function of these antibodies and how they protect us from the disease conditions. Before going into these antibodies types, let's start up what exactly an antibody is. Antibody is actually a protein or a polypeptide chain which is a high molecular weight protein. These proteins are produced by specialized B cells which are stimulated when a antigen or a pathogen intrude our body. So these antibodies are produced in our body only when there is a antigen or pathogen which intrudes our body. So subsequently they are produced and they invade out all these pathogens which are formed in our body and they neutralize and destroy these pathogens from our body. Now, how exactly an antibody looks like? The antibody has been said that it looks like a structure of Y letter. So there is a stem and there are two branches which are there, and on these branches there are antigen binding sites which are present to which this antibody, with the help of which these antibody binds to the antigen. And these antibodies are very specific to the antigens bindings. So these antigen binding sites are very specific and they are being recognized when the specific antigen is present in this immune surveillance system. So the antibody specificity is one of the major characteristic feature of the antibody. So the antibody will react only with the cells or the antigens or the pathogens which have got a complementary structure with the antigen binding site. So various types of antigens are shown over here with various geometrical structures, and we can see only the antigen which is having the complementary structure with the antigen binding site is getting bound to it. So these antibodies are very specific and specialized molecules which are circulating in the immune system. So wherever they recognizes or interferes with a antigen, they immediately recognize that particular thing and they get rid of this thing by neutralizing and activating the immune response system subsequently. So before going into this antibody, let's take up what exactly a immune system cells are. The immune system, we have already discussed, the immune system cells are majorly broadly divided into two major categories. The first one is called as the humoral system, and the second one is called as the cell-mediated immune response system. The humoral system is basically comprised of the B cells which contains the antibodies bound to this membrane of these particular cells. So these are particularly antibody bound cells in which these antibodies acts as a receptor. While the cell-mediated immune response system mainly comprise of the T helper cell and the T cytotoxic cell. As the name is suggesting, B cells are the cells which mature up and subsequently grow up in the bone marrow, while the T helper cell and the T cytotoxic cell, which is shown in the pictures, are generally developed and growing in the thymus organs in our body. In the picture over here, you can see the electron micrograph, which shows the exact pictures of how exactly we can see the immune system cells within our body. So the first picture is the small lymphocytes, which are basically the T cells or the B cells. Subsequently, they mature to form the blast cells, and we can see the size of the cells increase down, and subsequently when it gets interacted with the antigens, they form the plasma cells or the B cells, which finally goes out to synthesize the antibodies which are secreted on the surface of it. Now, the human body is having a human lymphoid system. So this human lymphoid system plays a major role in interacting, neutralizing and destroying all the pathogens or the antigens which invade into our body. So whenever there is a pathogen or an antigen which invade our body, or some cells within our body gets disrupted or virus infected, this immune system or the lymphoid system plays a major role in inactivating or destroying these particular pathogens or the antigen. The lymphoid system is basically divided into two major systems. The first one is called as the primary organs of this system, and the second is called as the secondary organs of this system. The primary organs comprise of the thymus and the bone marrow. So the thymus and the bone marrow are the organs which are responsible for the development and the maturation of the B cells. If the B cells matures, then they are developing into the bone marrow organ, while if the T helpers or the T cytotoxic cells are developing, so they are developing in the thymus cells. So we can see in the diagrammatic representation, there is a thymus which is red in color. It is just above the heart, and there is a bone marrow which is shown. So the femur bone is shown over here, in which the B cells are matured up. Now subsequently, there are secondary organs which are present throughout our body, of which the main organs are the spleen, tonsil, adenoids, then the lymph nodes comes. So all these secondary organs are connected to one another with the help of the lymphatic vessels which run throughout our body. So we can see purple color lymphatic vessels which are running throughout our body. So with the help of these lymphatic vessels and the blood vessels, these lymphocytes are circulated throughout our body. So these lymphocytes or the antibodies or these immune response cells are circulated within the body plasma circulation or through the lymphatic system throughout our body. So in our body, wherever there is an intrusion of these antigens or the pathogens into our body, these cells can immediately recognize these particular pathogens or the antigens and can neutralize and destroy them by activating the immune system cells in the subsequent process. So let's come into the B cells, which are of the main importance for as far as the antibody secretion or the productions are concerned. The B cells are majorly matured up or formed in the bone marrow cells, which are shown in the picture. They were present in the bones. So in the bone marrow cells, these cells develop, and these develop through the stem cells. So the stem cells go on to form the B cells, which have now become a committed B cells to grow into form the next level of the primary B cells, which are the B memory cells or the B effector or the plasma cells. So subsequently the B cells are formed. Then the B cells are, when it matures up in the bone marrow, it is transferred to, with the help of the lymphatic vessels, to the various lymph organs or the lymph nodes, where exactly the antigen antibody interactions takes place. So over here, as soon as these B cells interact with the antigen, these get into the excited form and start forming the memory B cells or the effector B cells or the plasma cells. So if you can see the picture over here, there is a clonal proliferation or clonal selection of B cells which are characteristically specific for that particular antigen which has been primed to this B cell. So these B cells recognize these antigens and start developing or forming the antibodies which are very specific to these antigens in the subsequent invasion processes. So subsequently the B memory cells are produced, as well as B effector or the plasma cells are produced. So the plasma cells are the cells which are responsible for the secretion of the antibodies into the blood circulation or the blood serum. So these plasma cells are the one which are secreting the antibodies continuously into the blood serum so that they can be circulated through our body. So we can see over here there are plasma cells which are secreting the antibodies in the blood serum, so they are circulating through our body. And secondly, there are certain memory cells which are present, which are very necessary for the immunologic response. Whenever these antigens subsequently invade our bodies, there are memory cells which are present in our body. They immediately recognizes these viruses, bacteria, or the antigens, and immediately activate to produce the antibodies for the subsequent clearance and the destruction of these particular pathogens from our body. So these antigen recognize maturation, and the antigenically committed B cells then subsequently proliferate and differentiate to form the plasma and the memory cells, which plays a major role in the lymphatic systems, thereby neutralizing and destroying these antigens or the pathogens in the subsequent by eliciting or evoking the immune response cells. So the antibody by itself doesn't clear or destroys these antigens or the pathogens, but what it does is it immediately recognizes these antigens or the pathogens, it binds to it, subsequently we will see it activates a number of pathways which are responsible for the immune system activation, and subsequently what happens is as soon as these immune system responses are activated, they come into the picture and they immediately clears out and destroys these antigens from the blood circulation or from the body. So if you can see the picture over here, it is a picture which is showing the interaction pattern of an antibody which is on the left hand side and an antigen which is on the right hand side. So we can clearly see an antigen antibody interaction and how exactly it happens in our body through a complementary interaction pattern which is shown over here. Now, before going into the antibodies, let's take up what exactly an antigen means, or what is an antigen. An antigen is a molecule which can be a foreign particle, protein, foreign protein, virus, bacteria, parasite or fungi which invades our body or gets into our body by various processes. So as soon as they get into our body, they are recognized as a foreign particle by our immune system, which is circulating with the help of the lymphatic vessels and the blood circulation. So they recognizes them, and what happens is as soon as these antigens get interacted with the B cells, they get excited to form the effector or the plasma cell, which subsequently secretes up the antibodies into the blood circulation, as well as they go on to activate the memory B cells which remains in our body circulation. And whenever there is a second antigen attack into our body, these memory cells come into the picture. Immediately they recognize these particular antigens which have already invaded our body, and they activate the plasma cells and subsequent pathways for the immune clearance. And these antigens also activate the B cells, which goes on to form the effector T cells, which can be C-type cytotoxic cells or the T helper cells, which also plays a major role in the immune clearance, and subsequently they also activate to form the memory T cells which remains in the blood circulation. So these antigens are basically the immunogenicity property which elicit the immune system to get activated, and as soon as the immune systems get activated, they immediately recognizes these antigens which is circulating in our body or which is getting stationed at one particular part of our body. They immediately recognize them and they come into the picture at that particular location with all the lymphocytes and the lymphatic cells into the picture, and they get these things into the immune clearance thing, and they neutralize and destroy these antigens of the pathogens from our body. Now, what are the various mechanisms through which these antibodies clear these antigens or the pathogens from our body. There are three major mechanisms by which this antibody clears these antigens from our body. The first is called as the opsonization, the second one is called as the complement system activation, and the third one is called as the antibody mediated cell cytotoxicity clearance. Well, we come into each of them one by one. Now the first one is called as the opsonization. So what happens is, as soon as the pathogen or the antigen which intrude our body, it is being recognized by the antigen. So these antibody recognizes these antigens or the pathogens, and what they do is they go and attach to these particular antigen. And subsequently what happens is they activate certain neutrophils and the macrophages into the particular area. So these macrophages and the neutrophils are activated. What happens is subsequently these particular antigen antibody complex goes on to attach itself to these macrophages with the help of the Fc receptors which are present on the surface of these macrophages. So the Fc receptor are the receptor which are basically designated for the attachment of the constant region of the antibodies to these macrophages or the neutrophils. So as soon as it binds to it, it phagocytizes these antigen antibody within itself, and subsequently it neutralizes by various chemical or the enzymatic processes and destroy these particular antigens or the pathogens from our body. So if you can see the picture over here, I have shown that how this antigen and antibody interact, and how after interaction with the antigen antibody, how it goes and binds to the Fc receptors in this particular macrophages, and after the binding with the Fc receptors in these macrophages, they are opsonized or phagocytized within these macrophages, and subsequently what happens is, with the help of the enzymatic process, they are destroyed from this particular system. Now the second major pathway through which these antibody functions is called as the complement system. So the complement system are basically the glycoproteins which are circulating within our blood serum. So these complementary system are majorly of different various types and various structures. So what happens is, as soon as these antigens invade our body, these antibody recognizes these antigens and they bind to it. By binding to it, it subsequently activates the complementary system which is circulating within our blood circulation. So this complementary system is activated, and with the help of various pathways of complementary activation, what happens is this complementary system forms certain membrane attack complexes on the surface or the membrane of these pathogens. So suppose there is a bacterial or a viral infection which has infected our body. So what happens is they are immediately recognized by the antigens, and as soon as they are immediately recognized by the antibodies, they subsequently activate the complement system. The complement system come into the particular area, it gets accumulated over here by the various activation pathway, and finally what is formed is a membrane attack complex which is formed, which is called as a MAC complex, and this MAC complex is formed on the surface or the membrane of this particular bacteria or the virus. So there are perforations or holes which are formed in the bacterial or the viral membrane, and which causes instability in these bacterial or the viral system, and subsequently what happens that these bacteria or the virus systems dies because of the instability which is caused due to the membrane attack complex which perforates the membrane which are formed on the outer side of its surface. So if you can see the picture over here, there are certain membrane attack complexes which are shown over here, and what we can see is over here, with the help of this membrane attack complexes, it forms certain pores within this membrane, and as these pores are formed within the membrane, the bacterial or the viral systems get instabilized, and due to this instability of the membrane, there is a non-functioning of this particular viral or the bacterial part, and subsequently what happens is the complementary system comes into the picture with the help of the antibodies to get rid of these antigens, and what it leads to is the death of the pathogen. The third major process by which this antibody elutes out or destroys this pathogen is through the antibody dependent cell mediated cytotoxicity or ADCC. So with the help of the ADCC process, the antibody kills these pathogens or the antigens which has intruded our body. In our body, the immune system contains some specialized cells which are called as the NK cells or natural killer cell. So what happens is, as soon as the antigens or the pathogens invade our body, they are being recognized by these antibodies. And these antibodies, what they do is, they go and bind to these target cells or the virus infected or the bacterial infected cells, and they attaches these particular cells with the help of the natural killer cells, with the help of the surface receptors which are present on the NK cells, and subsequently these NK cells go about to kill and neutralize these antigens, and subsequently it causes the death of these pathogens or the target cells which are being infected with the virus or the bacterial things. Now, the antibody structure discovery started way back in early 1960s by two major scientists who were named Altman and Porter. They were instrumental in discovering this antibody structure, and for their pioneer work or the breakthrough work of discovering the antibody structure, they were awarded the Nobel Prize in the year 1972. So Altman and Porter were the one who were awarded the Nobel Prize for the discovery of these antibody structure. So what they said in their award ceremony speech is, our body is having about 50,000 different types of antibodies. Each of them are specific to a specific antigen. And what they said is, these antibodies appear in a shape of a Y-shaped structure which contains a stem at the bottom position and two angled branches which are present on the top position. Each of these branched chains are made up of one light and one heavy chain, and subsequently the stem is made up of remaining part of these heavy chains. We will come into the description of these structural details which they gave. The next major discovery was done in the year 1984 by three major scientists, Journey, Kohlherr and Milstein. They were instrumental in discovering and giving the various theories for the development and the maturation of the immune systems and how exactly these monoclonal antibodies are formed and developed. For their pioneer work in the year 1984, they were awarded the Nobel Prize, for which they discovered many theories which were concerning the specificity in the development and control of the immune system and the discovery of the principle for production of monoclonal antibody. Now, in their award speech, what they said is, the immune system is just like a gigantic computer which is connected to a number of communication networks through a different components. So the same thing happens in our body. The immune system is connected with the help of a number of lymphatic vessels and the blood vessels, and in our body there are about 10 to the power 12 immune system cells which are continuously circulating, and they produce billions of different types of antibodies. So we can just imagine how enormous and diverse forms of antibodies are produced within our body to target each and every different form of antigen which invade or which gets into our body. Now, there were different experiments which were done to elicit the structural details of these antibodies. The first major experiment which was done with the help of the chemical and the enzymatic process for the discovery of the antibody structure was the use of mercaptoethanol. So the mercaptoethanol reduction and alkylation process was used, and what it did is, whenever these antibodies was incubated with the mercaptoethanol reduction, what happened is they found that there were 4 polypeptide chains which were formed. Of these 4 polypeptide chains, 2 polypeptide chains were identical of 500 molecular weight chains, while 2 were low molecular weight of about 25,000 molecular weight chains. So they said that the two heavyweight chains as the heavy chains, while the two low molecular weight of 25,000 molecular weight chains were designated as the light chains. So through this experiment, they deciphered out that the antibody contained 4 different chains, and of these 4 chains, 2 are called as the heavy chains and 2 are called as the light chains. So the antibodies are a heterodimer which contains light as well as the heavy chains of different molecular weight. So this was a very fundamental and instrumental experiment which gave the next level of experiments in which the antibodies structure or the fine details of the antibodies were discovered. The next experiment which was done was with the help of the papain digestion. Papain is an enzyme. What it does is, as soon as these antibodies were incubated with the papain, the scientists found that there were 3 major fragments which were eluted out from the solution. Of these 3 major fragments, the 2 fragments were called as the FAB fragment or fragment antigen binding fragment. So these are the fragments which are responsible for the antigen binding sites. So these are the fragments which contains the antigen binding sites into them. Subsequently, what they found is, they found a FC region which is called as the fragment crystallizable region, or which is a part of the heavy chain constant region. As it was called as the fragment crystallizable because it was found that during the cold storage of the sample, these particular fragment crystallized. So that's why the name came in, that the FC fragment. So what they said is, due to this papain digestion, there were 3 fragments which were formed, of which 2 were identical, which were called as the FAB fragment or fragment antigen binding fragments, which contains the antigen binding sites onto it, and the 3rd fragment was called as the FC fragment, which was of about 500 molecular weight size, and which is the fragment of the heavy chain. Now the 3rd major experiment was done with the help of the pepsin digestion, which is also a proteolytic enzyme. So by the pepsin digestion, what the scientists found is, there was a FAB fragment, but 2 FAB fragments were combined together, so it was named as FAB all to the power 2. So these FAB 2 fragments were formed which binds to this antigen. So these fragments are nothing but the 2 FAB like fragments which are joined together with the help of the disulfide bonds. So, and along with this fragment, some of the multi-fragment parts of the FC region was also found, but the FC fragment cannot be recovered in total because the pepsin digestion, what it does is, it digested this FC region into a multiple fragment. So this pepsin digestion also gave up the scientists that there is a FAB2 region which is there, which combines both the FAB regions, and both these are interconnected with each other with the help of the disulfide linkages. So all these experiments over the period were instrumental in giving up the fine structural details of the antibody. So the basic antibody structure comes up that the antibodies are basically heterodimer, which means that it is made up of 2 basic units. The one is called as the light chain polypeptides, the other one are called as the heavy chain polypeptides. So there are 2 light chain polypeptide chains which are present and 2 heavy chains peptide chains are present. So that's why the name is coming heterodimer. So there is a dimer and both these dimers are different, that's why this is a heterodimer, and each of these light chains are attached to the heavy chain with the help of the disulfide bonds, and subsequently the interactions are taking place with the help of the non-covalent interaction. Let's come into the structural details of this particular antibody. So the very basic structure over here, if you can see, there is a heavy chain which is present and there is a light chain which is orange in color. At the top of both the heavy and the light chain, there is an antigen binding site which is present, and this site corresponds to variable region of light chain and the heavy chain. So variable region is the one which is responsible for the antigen binding in both light chain and the heavy chain. Subsequently, if I can see that the light chain is attached to the heavy chain with the help of a disulfide bond, and this heavy chain is running to the bottom, and there are two subsequent domains or the units which are formed, and these heavy chains are attached to each other with the help of again another set of disulfide linkages. So the disulfide linkages are playing a major role in first connecting the light chain with the heavy chain, and subsequently connecting both the heavy chains to each other, and this is helping up to form the antibody structure. So the antibody structure is having a variable region which is present on the top portion, which is responsible for the antigen binding, while the subsequent regions are called as the constant region, which are responsible for the various other biological activities. Now, the basic structure of the antibodies said that the first 110 amino acids of both the light and the heavy chain amino terminal part are called as the variable region, because there is a variability of the sequences. So there is a highly variable sequence which is present. That's why the variable region of the light chain is called as VL, which is variable region of light chain, while the variable region of the heavy chain is called as the VH, which says that variable region of the heavy chain. And the region of relative constant sequences beyond this variable regions was called as the C region or the constant region. So there are two regions. The first one is the variable region, where the sequence are variable and highly variable sequence are present over here. The second part is called as the constant region, where the sequence is constant over here. Now, the constant region of the light chain is called as CL, while the constant region of the heavy chain is called as CH. And the amino terminal positions of the variable region are the one which are responsible for the antigen. So I've just shown you the picture in which there were antigen binding site which were present in the variable region. So if we can see the structural details of these antibodies over here, we can see that there is a light chain which are present on the side, and there is a heavy chain which is running at the center. So the light chains are attached to the heavy chain with the help of the disulfide linkages, and subsequently they are having the variable light chain region and the constant light chain region, while the heavy chain is having the variable heavy chain region, which is green in color, and rest of the purple color is the constant heavy chain region which is present. Along with many of the antibodies are having a hinge region, which is the point where the attachment takes place with the help of the disulfide linkages. If we can see over here, there is an attachment of a carbohydrate moiety also along with this antibody structure. So it is said that this carbohydrate structure gives the extra solubility characteristic to these antibodies. Now, the subsequent work which was done on these antibodies was the sequencing of the amino acids of these antibodies. So the amino acid sequences were taken out, and it was said that the first amino acid sequence were done for the light chain sequencing. So the light chain sequencing resulted in finding out the constant and the variable regions in these particular chains. So in the light chain there were variable region of light chain and there was constant region of the light chain, and subsequently in the constant region there were two basic amino acid sequences which were found, which was called as kappa and lambda sequences. So in the human 60% of the light chains are kappa, while the 40% are lambda sequences. Now the second major work was done on this sequencing of the heavy chain. So the heavy chain sequencing characterized or gave the variable region which is called as the VH variable region, while subsequent remaining constant region revealed 5 basic sequence pattern which were corresponding to 5 different heavy chain constant region, which are mu, lambda, alpha, delta and epsilon. So there were 5 basic sequence pattern which was found in the constant region, and on the basis of these 5 basic sequence pattern, the antibodies were classified into 5 major classes of antibodies, and they are called as the isotypes. So these heavy chain constant region sequences gave up the class of the antibodies, and the antibodies are now classified into 5 major categories. The first one is called the IgM, which contains a mu kind of heavy chain region, while the second one is called as IgG, which contains a gamma heavy chain constant region. Then comes IgA, which contains alpha heavy chain constant region. Then comes IgD, which contains delta heavy chain constant region, and the final one is the IgE, which contains epsilon heavy chain constant region. So all of them were varying on the sequences of the constant region of the heavy chain. Now, as we classify these antibodies, there were 5 classes. The first one was IgG, which contains the heavy chain, which was gamma. So there were 4 major subclasses which were found, which were called as gamma 1, gamma 2, gamma 3, and gamma 4. And it can contain a light chain, which can be a kappa or lambda light chain. Then there was IgM, which contains a mu heavy chain. Then came the IgG, which contains a alpha heavy chain, IgE, which contains a epsilon heavy chain, and IgD, which contains a delta heavy chain. Now, these amino acid sequences or the polypeptide sequences were found to be folded and present in these antibody structure. So what the scientists found is these immunoglobulins of the heavy and the light chain showed some characteristic homologous units of about 110 amino acid residues. So they named these units as domains. These domains were nothing but a beta pleated sheet in which 2 anti-parallel beta strands were running in the opposite direction to each other, and they were stabilized with the help of the hydrogen bond and the disulfide linkages. So if you can see the structure over here, the beta pleated sheets contain 2 anti-parallel beta strands which are running in opposite direction, and both these polypeptide chains are stabilized with the help of the hydrogen bonding. So this secondary structure of the beta pleated sheets was further folded into the immunoglobulin fold, which contains a sandwich of 2 beta pleated sheets which are stabilized with the help of the hydrophobic interaction as well as with the help of the disulfide linkages. So there were different domains which were present in each of the light and the heavy chain. So the scientists said that the light chain has got 2 domains. The first one is called as the variable region domain of the light chain. The second one is called as the constant region of the light chain, while in the case of the heavy chain there was a variable region domain of the heavy chain, while there were 3 constant region domain present in the antibodies IgA, IgD, and IgG, which contained hinge region, while some of the antibodies they did not have the hinge region. Instead they had an extra constant region which was present. So both the heavy chain and the light chain were having the first variable domain, and subsequently there were about 3 or 4 constant region domains were present in the heavy chain depending upon the classes of the antibody, while the light chain contained only a single constant region domain into it. So based on this structure, the unified or the immunoglobulin fine structure were deciphered out, and it was instrumental in the functioning of these antibody functions. Now, if you can see the pictures over here, this is a ribbon kind of interaction which is showing the constant region of both the heavy and the light chain. So the heavy chains are being represented by the blue and the yellow ribbons, while the light chains is being represented with the help of the red ribbon. So if you can see, the N-terminal regions of both the heavy and the light chains are the variable regions, while rest of the domains are the constant region domains of the light chain and the heavy chain. And subsequently it was found that in many of the antibodies, especially in the antibody IgG, IgD, and IgA, there was a presence of a hinge region. The main function of this hinge region was to provide flexibility to these antibodies, while in the case of IgE and IgM there was absence of hinge region. Instead there was a presence of an extra constant domain which was present. These hinge regions were majorly made up of two major proteins which were proline and cysteine. So the hinge regions are the one which are responsible for giving extra flexibility to some of the antibodies. So if you can see the structure over here, we can see the interaction or the association pattern of the different domains of the variable regions of the heavy chain and the light chain, while the constant region of the heavy chain and the light chain interactions can be also seen, and there is a carbohydrate moiety which gives the extra solubility characteristic to these antibodies. Now we come into the antibodies, all the 5 classes of the antibodies one by one. The first class was called as the IgG. IgG is the most abundant antibody which is found in the serum, and 80% of the total serum immunoglobulin constitutes this one. IgG molecules contains 2 heavy chains which are of basic sequence of gamma, and 2 light chains which can be a kappa and delta light chain. There are 4 major subclasses of IgG which are IgG1, G2, G3 and G4. They all differed in subtle differences in the amino acid sequences as well as the number of the disulfide linkages which are present in them. So let's take up the first basic structure of IgG. So if we can see over here, there is a variable region light chain and the variable heavy chain region which is present, and there are constant light chain and constant heavy chain regions which are present, and at the hinge region both these heavy chains are attached to one another with the help of the disulfide linkages. So the hinge region is providing extra flexibility to this IgG molecules. Now let's come into the different subclasses of the IgG molecule. There are major 4 subclasses of IgG which are IgG1, G2, G3 and G4. All of them differ depending upon the number of disulfide linkages which are present. IgG1 contains 2 disulfide linkages, while IgG2 and G4 contains 4 disulfide linkages. IgG3 contains 11 disulfide linkages. So if you can see the structure or the figure over here, we can clearly see that the IgG is having 2 disulfide linkages, IgG2 is having 4 disulfide linkages, IgG4 is also having 4 disulfide linkages, and IgG3 is having 11 disulfide linkages. All these IgG subclasses are having different biological activities. So many of them are activating the complement immune system, many of them are activating the opsonization process, and many of them are taking part in the passive immunization. So we come into the biological activity of all these subclasses one by one. So the first biological activity, major biological activity of IgG is the passive immunization. So we all know that the passive immunity to the developing fetus from the pregnant mother is given by the transfer of IgG antibodies through the placenta. So IgG1, G3 and G4 antibodies readily cross the placenta and plays an important role in protecting the developing fetus. IgG3 plays a major role in complement activation, while the IgG1 and G3 are the one which are responsible in activating the opsonization process. So all these IgG3 and G1 are the major immunoglobulin G which are responsible for attacking the antigen or the pathogen which invades in our body. Now we come into the next antibody class which is called as the immunoglobulin M. So immunoglobulin M constitutes to about 5 to 10% of the total serum concentration, and the monomeric IgM weighs about 1,80,000 molecular weight, and it is generally expressed on the surface of the B cells. So they are membrane bound antibodies which are present on the surface of the B cells, while as soon as they are secreted into the plasma, what happens is they get into a structure which is called as a pentameric structure. So the monomeric IgM gets into a pentameric structure with the help of a J polypeptide chain. So if we can see the structure of the IgM over here, there are 5 monomeric unit of IgM antibody which are attached to one another with the help of the disulfide linkages, and there is a presence of J chain which is basically joining chain polypeptide which is helping to polymerize all this pentameric unit of immunoglobulin M. So immunoglobulin M is the first responder to any of the antigens which invades our body. So immunoglobulin M plays a major role in classifying the antigens and then get rid of these antigens from our body. As we saw the structure of the IgM, there are 10 antigen binding site, but because of steric hindrance, only 5 or fewer molecules of the antigen binding site work simultaneously. IgM is the first immunoglobulin which is produced in response to the antigens, and IgM has been found much more efficient than the IgG in activating the complement system. So IgM plays a major role in getting rid of these viral particles and all those things by activating the complement and the other system, immune response system. The next category of the immunoglobulin is called as the IgA, which constitutes to about 10 to 15% of the total immunoglobulin. This is majorly called as the secretory IgA molecule because it is majorly found in the external secretions such as the breast milk, saliva, tears, mucus of the bronchial, genitourinary or the digestive tracts. As we know that the first entry of any of the pathogens into our body is through the mucous membrane which are lining the surfaces of the organs. So these IgA secreting plasma cells are majorly lined on these mucous membrane cells, and they are produced from that particular plasma cells over there. So the secretory IgA having a different structure from the IgA, which are generally present either in the dimeric form or the tetramers. So if you can see the structure over here, there are dimers or two monomeric units of IgA which are attached to one another with the help of the J chain, which is the similar chain which is present in the IgM, and they are attached with the help of the J chain with the help of the disulfide linkages. So the constant region of the IgA monomer is attached to the J chain, and subsequently the J chains are also attached to one another with the help of the disulfide linkages. The secretory IgA generally has got an additional segment which is called as the secretory component, which combines both the constant region of the IgA, J chain and the other monomeric unit of IgA. So the secretory component is a very important component for the secretory IgA. As we have discussed, the IgA is present generally in the external secretion, so they provide an important line of defense against the bacteria such as Salmonella typhi, which causes the typhoid, Vibrio cholerae, which causes the cholera, viruses such as polio, influenza. And we have just discussed that the IgA is majorly present in the breast milk, so it is providing a protection to the newborn against various infections during the first month or the first year of the life. So the IgA antibody is one of the major antibody which is playing a major immune system mechanism. The next one is called as the IgE, which plays a major role in the hypersensitive reaction. So wherever there is an allergic or hypersensitive reaction, this IgE antibody plays a major role. And it was first discovered in the year 1921 by the two scientists, Prosnitz and Kutzner, who for the first time demonstrated the allergic reaction which was mediated with the help of the IgE antibody. So the IgE antibody is having a structure like this, which contains 4 constant region because there is an absence of the hinge region, and both these heavy chains are attached to one another with the help of the disulfide linkages. So all of us are aware of the asthma and the allergic reaction. So over here, if you can see, the asthma and the allergic reactions are a major public health problem. So many of us are allergic to the food sources such as peanuts, almond, cashew nuts, walnuts. So many of the foods or the food items which contains these kind of things, when we eat them, we get allergic reactions in our body. While in the case of the asthma, many of are asthmatic patient or have asthmatic patients in our home. So these are basically the chronic disease of the lung which causes inflammation of these lungs, and the patient suffers from in difficulty in breathing. So this is majorly caused by the environmental antigens or the infections which we breathe in. So over here I have shown an example how this IgE antibodies plays a major role in this asthma. So the asthma is majorly caused by the allergen, which is a ragweed pollen. So when we inhale these ragweed pollen, they go into the immune system, to the lymphatic vessels, they go to bind to these B cells. As soon as they bind to the B cells, they activate the B cells to form the plasma cells. These plasma cells start secreting the IgE antibodies. So these IgE antibodies go and attaches itself to the mast cells which are present in the circulating or the immune system cells. Now, as soon as these IgE antibodies goes and binds to these mast cells, they come up. They also bind with the pollen, and these pollen activates these mast cells to release the histamines or the other pharmacologically active or the allergic molecules into the circulation. So these causes many a times the asthmatic attack, running nose, sneezing, fever. So lots of symptomatic conditions start taking place. So IgE is one of the major antibodies which is playing a role in these allergic and the asthmatic attack condition. The next major class is the IgD, which is present at a very low concentration. It is present as the membrane-bound immunoglobulins which are expressed on the mature B cells, but no clear biological function of these IgD has been found, and the basic structure of the IgD is like this, which has got a hinge region and 3 constant region in the heavy chain and a 1 constant region in the light chain, while the variable region is through which it binds to the antigens. Now, before ending the things, the antibodies have been playing a major role in diagnostic, imaging and therapeutic reagents in the clinical medicines and identifying various diseases in our thing. One of the major use of these antibodies are now taking place with the help of immunotoxins. So these monoclonal antibodies are now used as the immunotoxins for, suppose for the tumor specific cells. So the tumor specific cells expresses certain antigens on the surface of it. So they have got tumor specific antigens present onto surface of it. So what the scientists are now doing is they are synthesizing the monoclonal antibodies which are specific to these tumor specific cells, and what they are doing is they are attaching a toxin cell, toxin chain or a toxin to these particular monoclonal antibodies, so that what happens is as soon as we inject them into the body, they can go into that particular tumor cells and get into these tumor cells, releasing the toxins from its surface, and these toxins can go and stop the translation or transcription process of these specific tumor cells. So these immunotoxins are now becoming the specific system for targeting the cancerous cells. So the antibodies have opened up the next level of research and the pharmaceutical work, so that they can be used in various diagnostic and the therapeutic uses in the future uses. So with this, I end my topic on antibodies. Thank you very much.
Thank you, sir. Thank you so very much for delivering such a nice lecture and making us aware of how antibodies work and what are different types of antibodies, about their structure and functions. Tomorrow also you will be with us, and tomorrow we are going to discuss on
antigen antibody interactions and how the various immunoassays are being developed on the basis of these antigen antibody interactions. So I will be talking about the antigen antibody interaction principles and the different immunoassays which are in the market for the general users and detecting the various disease conditions.
With this note, thank you, sir. Thank you so very much, and tomorrow also we are going to learn more through you. Thank you.
Thank you very much.